I can manage. You can hear me well?
Yes, madam. Madam, I think your computer audio is low. Could you increase that?
So we'll be waiting for some time.
No, madam, we will be going ahead because it's already time.
Your volume is still very low. I can't hear you well. Can you increase your volume by any chance?
My volume is high, madam. 1 second. Stop. Hello. Now?
No, it's the same, but it's okay. I'll have to manage, I think.
Now?
Yeah, slightly better.
Oh, yeah, fine. Given you the share option, you may share your PPT.
Okay. 1 minute. I'll just open. Can you see my screen? I think you're muted. You're muted. I can't hear you.
Madam, I can see.
Okay.
It is fine. It is fine.
Okay.
Go ahead, madam.
Okay, we can go ahead. All right. You can hear me clear, right?
Sure.
Okay.
Yeah.
All right.
So, at the outset, I sincerely apologize to all the attendees for delaying today's session by 7 minutes. Very sorry for the there was a technical glitch from the expert and my side. So, apologies to you, all of you, those who are waiting for the session. So, starting the WebEx webinar for today's topic is on drug discovery and drug development by Dr. Kirthana Sindhe from Merck Lab of Technologies Private Limited. I welcome Kirthana M. Sindhe to this webinar, and I also welcome all the students who are joining this webinar. Welcome once again, everyone. ಮೊದಲನೇದಾಗಿ ಇವತ್ತಿನ ಈ ಕಾರ್ಯಕ್ರಮಕ್ಕೆ ನಮ್ಮ ಜೊತೆಗೆ ಆನ್ಲೈನ್ ನಲ್ಲಿ ಯೂಟ್ಯೂಬ್ ನಲ್ಲಿ ನಮ್ಮ ಕರ್ನಾಟಕ ವಿಜ್ಞಾನ ಮತ್ತು ತಂತ್ರಜ್ಞಾನ ಅಕಾಡೆಮಿಯ ಅಧ್ಯಕ್ಷರಾದಂತಹ ಎಸ್ ಅಯ್ಯಪ್ಪನ್ ಹಾಗೂ ಮುಖ್ಯ ಕಾರ್ಯನಿರ್ವಾಹಕ ಅಧಿಕಾರಿಯಾದಂತಹ ಎಂ ರಮೇಶ್, ಆಡಳಿತಾಧಿಕಾರಿ ಮಹಾದೇವಗೌಡ, ಹಿರಿಯ ವೈಜ್ಞಾನಿಕ ಅಧಿಕಾರಿಗಳಾದಂತಹ ಡಾಕ್ಟರ್ ಆನಂದ್ ಆರ್, ಉಮೇಶ್ ಗಾಡ್ಗೆ ಮತ್ತು ಶ್ರೀನಿವಾಸ್ ಮತ್ತು ನನ್ನ ಎಲ್ಲಾ ಸಿಬ್ಬಂದಿಗಳಿಗೂ ಈ ವೆಬಿನಾರ್ಗೆ ನಾನು ಸ್ವಾಗತವನ್ನು ಬಯಸ್ತಾ ಇದ್ದೀನಿ. ಇವತ್ತಿನ ವಿಷಯ ಡ್ರಗ್ ಡಿಸ್ಕವರಿ ಮತ್ತು ಡ್ರಗ್ ಡೆವಲಪ್ಮೆಂಟ್ ಎಂಬ ವಿಚಾರದ ಮೇಲೆ ಡಾಕ್ಟರ್ ಕೀರ್ತನಾ ಶಿಂಧೆ ಇವರು ತಮ್ಮ ವಿಚಾರವನ್ನು ತಿಳಿಸಲಿದ್ದಾರೆ. ಅವರಿಗೂ ಈ ಕಾರ್ಯಕ್ರಮಕ್ಕೆ ಮಗದೊಮ್ಮೆ ಸ್ವಾಗತವನ್ನು ಬಯಸ್ತಾ ಡಾಕ್ಟರ್ ಕೀರ್ತನಾ ಅವರ ಬಗ್ಗೆ ಕಿರು ಪರಿಚಯವನ್ನು ನಿಮಗೆಲ್ಲರಿಗೂ ನೀಡಲು ಕೊಡ್ತಾ ಇದ್ದೇನೆ. ಡಾಕ್ಟರ್ ಕೀರ್ತನಾ ಶಿಂಧೆ ಅವರು ತಮ್ಮ ಪೋಸ್ಟ್ ಡಾಕ್ಟರ್ ರಿಸರ್ಚ್ ಅನ್ನ ಕ್ಯಾಲಿಫೋರ್ನಿಯಾ ಸ್ಯಾನ್ ಫ್ರಾನ್ಸಿಸ್ಕೋ ಯೂನಿವರ್ಸಿಟಿಯಿಂದ ಪಡೆದಿದ್ದಾರೆ. Anti-malaria drug discovery and target identification in collaboration with Pfizer Pharmaco ಟೈಟಲ್ ಮೇಲೆ ಅವರು ಪೋಸ್ಟ್ ಡಾಕ್ಟರಲ್ ರಿಸರ್ಚ್ ಅನ್ನು ಮಾಡಿದ್ದಾರೆ. ತಮ್ಮ PhD ಪದವಿಯನ್ನು JNCSR ಇಲ್ಲಿಂದ ಬೆಂಗಳೂರು ಇಲ್ಲಿಂದ ಮಾಡಿದ್ದಾರೆ. ಅದರ ಟಾಪಿಕ್ ಬಂದ್ಬಿಟ್ಟು ಮಾಲಿಕ್ಯುಲರ್ ಬಯಾಲಜಿ ಅಂಡ್ ಜೆನೆಟಿಕ್ಸ್. ಇದು 2014 ಅವರು ಕಂಪ್ಲೀಟ್ ಮಾಡಿದ್ದಾರೆ. ಕಂಪ್ಲೀಟ್ ಮಾಡಿದ್ದಾರೆ. ಅದು ಅಲ್ಲದೆ 2009ರಿಂದ ಈವರೆಗೆ ಸತತ 11-12 ವರ್ಷಗಳು ತಮ್ಮ ವರ್ಕ್ ಎಕ್ಸ್ಪೀರಿಯನ್ಸ್ ಅನ್ನ ಹೊಂದಿದ್ದಾರೆ. ಇದು ಅಲ್ಲದೆ ಅವರ ಸ್ಪೆಷಾಲಿಟಿ ಏನು ಅಂತ ಹೇಳಿದ್ರೆ ಅವರು ಹಲವಾರು ಟೆಕ್ನಿಕಲ್ ಸ್ಕಿಲ್ಗಳಿಗೆ ಪ್ರಖ್ಯಾತರು. ಅದರಲ್ಲಿ ಪ್ರಮುಖವಾದವುಗಳು ಬಯೋ ಆಸಿಸ್, ಮೊಲೆಕ್ಯುಲರ್ ಬಯಾಲಜಿ, ಪ್ರೋಟೀನ್ ಅನಾಲಿಟಿಕ್ಸ್, ಬಯೋ ಇನ್ಫಾರ್ಮ್ಯಾಟಿಕ್ಸ್, ಸೆಲ್ ಬಯಾಲಜಿ, ಜೆನಾಮಿಕ್ಸ್ ಮತ್ತು ಇತರೆ ಸ್ಕಿಲ್ಸ್ ಗಳಾದಂತ ಸೈಂಟಿಫಿಕ್ ರೈಟಿಂಗ್, ಸೈಂಟಿಫಿಕ್ ಪ್ರೆಸೆಂಟೇಷನ್, ಎಲ್ಲ ವಿಜ್ಞಾನಕ್ಕೆ ಸಂಬಂಧಪಟ್ಟಂತ ವಿಚಾರಗಳಲ್ಲಿ ಅವರು ವೈಶಿಷ್ಟ್ಯತೆಯನ್ನು ಹೊಂದಿದ್ದಾರೆ. ಅದು ಅಲ್ಲದೆ VIT ವೆಲ್ಲೋರ್ ಇಲ್ಲಿಂದ ಅವರಿಗೆ ಸಿಲ್ವರ್ ಮೆಡಲ್ ಅನ್ನು ನೀಡಲಾಗಿದೆ. ಫಾರ್ಮಾಮೆಟಿಕಲ್ ಕೆಮಿಸ್ಟ್ರಿ ವಿಷಯದ ಮೇಲೆ ಅವರಿಗೆ ಮೆಡಲ್ ಬಂದಿದೆ. ಅದು ಅಲ್ಲದೆ UCSF ಸ್ಕೂಲ್ ಆಫ್ ಮೆಡಿಸಿನ್ ಇಲ್ಲಿ ಟೀಮ್ ಲೀಡ್ ಆಗಿ ಅವರು ಕಾರ್ಯನಿರ್ವಹಿಸಿದ್ದಾರೆ. ಪ್ರೋಗ್ರಾಮ್ ಕೋಆರ್ಡಿನೇಟರ್ ಆಗಿ ಅದೇ UCSF ವುಮೆನ್ಸ್ ಇನ್ ಲೈಫ್ ಸೈನ್ಸಸ್ ಇದರ ಸಂಸ್ಥೆಯಲ್ಲಿ ಕೋಆರ್ಡಿನೇಟರ್ ಆಗಿ ಕೆಲಸ ಮಾಡಿದ್ದಾರೆ. ಈ ಎಲ್ಲ ವಿಚಾರಗಳಲ್ಲಿ ಅವರನ್ನು ವೈಶಿಷ್ಟ್ಯಪೂರ್ಣವಾಗಿ ತೊಡಗಿಸಿಕೊಂಡಿದ್ದಾರೆ. ಹಾಗೂ ಅವರು ಪ್ರಸ್ತುತ ಮರ್ಕೋ ಪ್ರೈವೇಟ್ ಲಿಮಿಟೆಡ್ ನಲ್ಲಿ ತಮ್ಮ ಕಮರ್ಷಿಯಲ್ ಮಾರ್ಕೆಟಿಂಗ್ ಮ್ಯಾನೇಜರ್, ಕಸ್ಟಮರ್ ಎಕ್ಸ್ಪೀರಿಯನ್ಸ್ ಲ್ಯಾಬ್ಸ್ ಇದರ ಈ ಸಂಸ್ಥೆಯಲ್ಲಿ ತಮ್ಮ ಸೇವೆಯನ್ನು ನೀಡ್ತಾ ಇದ್ದಾರೆ. ಹೀಗಾಗಿ ಅವರನ್ನು ಈ ಟಾಕಿಗೆ ವೆಬಿನಾರ್ಗೆ ಸ್ವಾಗತವನ್ನು ಕೋರುತ್ತಾ ಮುಂದಿನ 45ರಿಂದ 50 ನಿಮಿಷ ಈ ಟಾಕ್ ಆಲಿಸಲು ಅವರನ್ನು ಸ್ವಾಗತಿಸ್ತಾ ಇದ್ದೀನಿ. ಮೇಡಮ್, ನಿಮಗೆ ತುಂಬ ಹೃದಯದ ಸ್ವಾಗತ. ಥ್ಯಾಂಕ್ ಯು.
Thank you so much, sir. Okay, we will get started.
Yes, madam, please go ahead.
Okay. Good evening, everyone. Myself, Kirthana here. You've already heard about my introduction. I would like to get started with the topic for today's talk, which is drug discovery and drug development. So currently, I'm a senior strategic manager at Customer Experience Lab for Merck. I would like to give you all a brief introduction about our labs and the company. So just give me a minute. So we are vibrant science and technology company. We are leaders in life sciences. We are a vibrant team of specialists in healthcare, life science, and performance materials. We're dedicated to solve the toughest problems in life science by collaborating with global scientific community. We have established 2 labs, which is called Merck Innovation Lab, located in Bangalore, and Merck High Skill Development Center, which is in collaboration with CSR Imtech in Chandigarh. So these labs are set up to enable and facilitate life science research in India, to provide tools and techniques to conduct researches, and also to upgrade scientifically, to focus on different various job opportunities for all the students, research scholars, and academicians in life science field. So we have different working areas in the lab. So if you see here, right from molecular biology, it goes up to proteomics. We provide a lot of skill development programs to the scientific community. We have our LinkedIn group, so feel free to use this link. I can also put it in the chat box. We are having this LinkedIn group where we keep posting a lot of upcoming skill development programs in life science, especially in biotechnology. So today's topic is drug discovery and drug development. So I would be first starting with the definition, so that you all are on the same page and you understand most of the concepts very clearly. So drug discovery, it is the process by which new pharmaceutical candidates are discovered. So that is how that's why it's called discovery, where you're finding out the new pharmaceutical candidate which can have an effect on the body or any kind of disease. Drug development, it is a process of bringing that new pharmaceutical drug, whatever you have discovered, you need to bring that to the market. So once you have discovered, the process is called drug development, when you bring that product into the market. So these are the two important definitions, and it is important for us to understand why do we need to understand drug discovery and drug development, because it's a complicated and an expensive process. You will see that it takes average of 10 to 15 years for a drug to reach into the market or a pharmacy. And why, and we should also be sure why it takes so long and why the process is so complicated. So if you understand about the process, it will specifically help for all the clinical researchers and also the company itself, because they invest a lot on a particular molecule that comes into the market. So the lot of investment goes into discovering a drug for an intended use, and also for it to be developed further so that the efficacy and safety is met when it's administered in patients. So it's very important for the company who invest in drug discovery and drug development for them to understand this entire process, and also for clinical researchers, because they are the ones who understand the target patients and these drugs are monitored. So, and as you all know, the drug should be safe and it should be useful for the target patients. So, this is about the definition and why this process is very important. And if you all know, there are different stages in drug development process. So, the first step, as I already mentioned, it is drug discovery. The second step is preclinical stage. Generally, you try to understand how the drug responds in animals. Third is the clinical trials, where you will test the drug in the humans. So there are again different phases within the clinical trial, starting from phase 0. And you have FDA drug review. That is, once your drug has passed all the clinical trials, the FDA will do the review of the clinical trials and how the drug is responding in the patient. So the FDA approves it and then it comes into the market. But that is not the end of the story. Even after the drug is gets into the market, there's something called post-market drug safety monitoring, or the post-surveillance survey that is being done for it to be monitored, that if any other side effects are possible. Because generally when you do clinical trials, it's done on large number of groups, especially in phase 3. Let's say 5,000 patients are tested, but our population is so huge, there's a possibility that certain population are having adverse reactions once it's been approved. So that's why the post market surveillance is done, so that if there is any side effects, it will be addressed, and if needed, the drug will be withdrawn. So these are the different stages of drug development process. Now, moving on to first to understand what happens in drug discovery. What are the different ways you can discover the drugs? So, most first, very important is any drug that you want to discover, you first need to know which disease against which you want to discover the drug, right? So, understanding the disease completely, and for which drug you want to use for the disease, that is what is most important in drug discovery. So as you all know, there's so many drugs which are in the market. Maybe if you let's say, for example, you're trying to discover a drug for a breast cancer. So you need to understand completely about breast cancer, which type of breast cancer, and what kind of already the drugs exist, right? So based on the existing drug, you can create certain variants. Maybe you can change a little structure in the drug and you can create a new molecule. So that becomes a variant. Or if you want to identify some biochemical pathway in the disease so that the molecule can act on those biochemical pathway, you can also get from a natural compound. Fourthly, you can repurpose. What is repurpose? That is, there's already a drug existing in market, but you're now going to use for the intended disease that you're looking for. For example, let's say you there is a drug which is used for autoimmune diseases, right? And that drug is approved. Now, maybe that same drug can work in cancer treatment as well. So you will use that same drug, but this time you need to get it approved for cancer. It is approved for autoimmune, but anytime you want to use it for a different purpose, therapeutic use, it has to be approved for that particular use. So that is why it is called repurposing. So that is about how are the different ways that you can discover the drug. And shown here are the different steps in drug discovery. As I mentioned, first is to identify a disease so that you know which drug can be identified or discovered for this disease, and find the potential targets. So all of you are aware about genomics and proteins. If not, I'm sure all of you know what is genes and what is protein. So if you know the central dogma of molecular biology, it is the DNA, then RNA and protein, right? So generally any disease targets, it is a protein, and if you go a little more at the molecular level, it is genes or the DNA. So it is important to see which is the gene or a protein this drug is targeting. So those are called potential targets, because you need to know for which your drug, which target it is going to work on. Otherwise you will not know how this drug will work in the body or with the targets. Then the third step is to screen all the compounds. Let's say you have some 10,000 compounds, a library of compounds are there, but you can't develop, as you already saw, one molecule, it takes 10 to 15 years, and investment is also very huge. So only one molecule can be pushed at a time, let's say for a time period of 5 to 10 years. Then once that molecule goes into the clinical phase, then you can invest further on the previous molecules. So basically what I'm trying to say here is a screening of compounds is very important. Out of those thousands of compounds, you need to come up with one or two which is more effective, which can bring the best outcome for the disease treatment. So that is how there are different ways you can do virtual screenings. What I mean by virtual screening is there are a lot of softwares available. You can do a lot of modeling and see how the structure of the compound fits with the target and how it can actually bring about an therapeutic effect. So that these days it's called chemoinformatics. You can do through softwares and have all of those structures of the compound and screen virtually. That is one option. And second one is high throughput screening, which is generally done in vitro, that is in the labs using different cell lines. So you can screen thousands of compounds again against the particular cells, and you can see if those cells are being killed with these compounds. So that is how you do a lot of screening of compounds once you know the target. And lastly, you need to find a particular drug. So either, as I already mentioned, by molecular modeling, that you will see how the drug is fitting into the target. The best fit will be chosen to give a great effect in the patient. So this is how you do the discovery. And there are different ways you can discover. We already mentioned about, you can do chemical synthesis, you can screen natural products, you can do target based design. That is, if you know the target, you can actually you can actually identify a drug that best fits with the target, and you can design those drugs. And also you can actually screen for solubility, stability, absorption, metabolism, and how the drugs are transported, how it interacts with the body. So let's see more deeper now how else you can do further drug discovery. So all of you, I'm sure you understand what is pharmacodynamics and pharmacokinetics. These are very two important terms that is used in drug development and drug discovery. So let me quickly tell you what it is. So pharmacodynamics, which is PD, which is that is the drug action and drug mechanism that happens in the body. So that is what is mean by pharmacodynamics. And in pharmacokinetics, you will understand how the drug gets absorbed, how it gets distributed in different tissues in the body, how it gets metabolized and excreted from the body. So let's understand another step deeper what these two things mean, because based on these, lot of drugs are selected for the drug development. So if you look into pharmacokinetics, what it means is what body does to the drug, right? So when you take a pill, any medicine, your body will also do something to the medicine. So that is what we is called as pharmacokinetics. Let's see what it does. So first is absorption, right? Once you take the medicine, it will get into the blood. It has to come into the blood because it is the blood that goes to all different tissues in the body. Because if you want any therapeutic effect, the blood has to carry the medicine to the particular site of action, and that's how it will help. So absorption of the medicine into the bloodstream is very important. So that is called bioavailability of the drug. So that's our absorption. Then metabolism, right? So sorry, you have distribution. That is how the drug goes into different tissues in the body. Let's say you want the drug to reach kidneys, right? So it has to get distributed in the kidney from the bloodstream. So that is called distribution. Metabolism is anything, I'm sure anything that you eat, right? Even the food when you eat, it gets metabolized, it gets broken down. So similarly, even the medicine gets broken down after certain time, especially in the liver. So that is called metabolism, which is again brought about by the body, especially liver. And excretion, right? That is how the drug gets removed or released from your body. So these are the four important processes which comes under pharmacokinetics, that is called ADME. So as you keep moving ahead in your career, you will understand more about pharmacokinetics of the drug, which is very important. Now let me give you a simple example of how why it is important, how the concentration of the drug happens in the body. So when you take a medicine, you will see that in the blood, so blood, it is drug plasma concentration, which means the concentration of the drug in your plasma, that is in your blood, you will be able to measure when you take. So if you see, this is a curve. Pharmacokinetics is measured this way. Why this is very important? So you will see that there are two lines drawn here. This is called a minimum effective concentration. That is minimum concentration of the drug that is needed to give any kind of benefit, right? Any small response that is needed. That is called minimum effective concentration. That is, this much minimum drug concentration is needed for you to respond for any particular disease. And here on the top, you will see is MTC, that is maximum therapeutic concentration, which means that, sorry, maximum toxic concentration. That is, if you have too much of the drug in your blood, it becomes very toxic. So you cannot have anything above this concentration. And hence you will see that this is called a therapeutic range. That is, it is the drug concentration should be within this range for you to get the best effect for the particular disease. So if it goes above MTC, then you will have very toxic effects. Lot of side effects will happen. If it is below MEC, that is, there is no much concentration there, so there will be no effect at all. So maintaining a therapeutic range for any particular drug is very important, and that is how it is being measured. So whenever you discover a drug, it will be tested for ADME, especially the drug plasma concentration, how it gets, how much concentration is available for it to have a therapeutic response. So this is about ADME. Now going into pharmacodynamics. So here, what a drug will do to the body. What we saw in pharmacokinetics is what does the body do to the drug. Now we are seeing what a drug will do to the body. So if you see any cells that we have, it has extracellular space, you have a cell membrane and intracellular environment. So there are a lot of receptors which is present on the surface of the cells. So when the drug molecule goes inside, it will bind to all these receptors on the cell membrane, and then whichever receptors gets engaged, there is a internal cascading signal that happens, and there is a biological response. So you will see that the drug plus receptor is called drug receptor interaction, and because of this interaction there will be a biological response. So This is called pharmacodynamics, and you will see that mechanism of action of drug is very important. What you mean by mechanism of action? That is, whenever a drug binds to a particular target, when it interacts, there will be a pharmacological effect. And any drug that you discover, it will fall only under three important categories. That is, it is called an agonist. It can be a mix, that is agonist and antagonist, and an antagonist. So let me quickly tell you what these means. So what happens in case of agonist? So if your drug is an agonist, it will bind to the receptor and there will be an activation in the cell pathway, right? So this is called full activation. That is, a drug will activate a signaling pathway. So some of your drugs can be an agonist. Let's see antagonist. That is, you need, when a drug binds to a receptor, there will be no activation. That is, you're blocking any kind of activation. In certain diseases, you will need some activation to be stopped. Otherwise, it becomes very, the activation only leads to a lot of different complications. So in those cases, you actually need an antagonist. That is, you want to block the response. So here you will see that antagonist will do not activate and it will block the response. And the third category, if you see, it is a mixed, that is an agonist and an antagonist. So this is more mainly if you need only partial activation of the signaling. In that case, this will be the best category to be used. So these are the different mechanism of action of the drug based on what kind of response you want internally, that is at the cellular level. And based on that, your drugs will fall under these three categories. So when you discover. And if you go more deeper into mechanism of action, I'm sure all of you know about this. This is a cell, and you know there is a cell wall, cell membrane. There are a lot of metabolic pathways that happens inside the cells. You have a nucleic acid synthesis happening here and protein synthesis happening here. So whenever you use any drug, it falls under again these five important mechanisms. So your drug can either inhibit the cell wall, right? So that is, if the bacteria doesn't have a cell wall, then it cannot survive on its own. So it needs cell wall, but when you have a drug that can inhibit the cell wall, the bacteria or any other pathogen would die. Similarly, if you stop protein synthesis, again, the bacteria cannot survive. If you stop nucleic acid synthesis, bacteria cannot survive. Metabolic pathways, like for example, folate biosynthesis, if you block your drug for metabolic pathways, the bacteria cannot survive. And lastly, cell membrane integrity, right? Cell membrane is also very important to maintain the structure of the bacteria. So these are various ways by which your drugs can act. It can either act on the cell wall, protein synthesis, it can block DNA synthesis, metabolic pathway, or the cell membrane integrity itself. So this is how most of the drugs will fall under different categories. So understanding the mechanism of action is very important before you take it to the drug development process. So now we understood completely why drug discovery is important, how you can do drug discovery, and what are the different parameters to consider in drug discovery when you're discovering a drug, right? Pharmacodynamics, pharmacokinetics, and what is its mechanism of action. So once you have all of that information and you have discovered a particular drug, now it's time for it to be brought to the market. So that is called the drug development process. So what do you do first? So once you've discovered the drug, you need to do preclinical testing. So preclinical testing, it can be in vitro, that is in the cell lines, or it can be in vivo, that is in the animal models, like let's say rabbit, mouse, rat, even monkeys to a certain extent, as they have a highest homology with human. So based on the preclinical testing, we will evaluate what all is evaluated here. You will evaluate efficacy, so toxicity. If the drug is toxic in animal model, it'll the animals will show a lot of side effects, it will die, or any other side effects can be identified here in the animal models. And I already mentioned the ADME testing, that is pharmacokinetics. So in animals, you will try to understand how the drug gets absorbed, how it gets distributed, metabolism, elimination, all of that information you're actually getting in the animal models. So this way, once you know in preclinical testing, your drug is working very well in vitro in the cell lines, it's killing the cell lines, you are evaluating all of these efficacy, toxicity, and ADME, then your drug is good to go. You also evaluate how much of the drug concentration is needed for to give a therapeutic response. So now your drug goes into phase 1 study. So phase 1 study, it happens inside the human beings. That is the first in human study that happens. And here the medication. So here you want what is done in phase 1 study is mainly efficacy and safety, right? And here it is done on a very small size population, only 20 to 100 patients you're going to do. And once you will administer the drug, and you will try to see if this drug is safe, whether it is also effective. So that is the goal in phase 1 study. Whereas in the phase 2 study, you have two groups, and now your population is increased to 100 to 500 people, which was earlier just 100, a small size. Now it is mid-sized population. And here, as I mentioned, there are two groups, and it is a double-blinded study. What do you mean by double-blinded study is the patients and the person, that is the researcher who conducts the study, both of them don't know that what is being administered to them. So a certain group is called control group, that is placebo. You are not giving the drug, but you're still injecting just the saline, where the patients would feel that they got some drug. So you will try to evaluate that's called the placebo effect. And also actual drug is given in the other group of patients. So now you have two different groups of patients, and it is a double blinded study. You'll try to again evaluate both safety and efficacy. So this is done in the phase 2 study. Whereas in the phase 3, you are now increasing the population up to 5,000. Again, it's a double blinded study, and now the phase 3 is very critical. You have to see what's its maximum efficacy and safety, and now you're closer to the approval of the drug. So FDA will now review all of your studies in from preclinical to all the phase 3 trials, and it will see how well your drug is doing. It will keep monitoring for 6 to 10 months. There's a routine inspection of all the clinical site studies that are conducted, and that's how you get the approval for the drug. And then the drug, as I mentioned, after being marketed, still you will have to monitor how this drug is doing well. And time to time the reports have to be published, has to be shared with the FDA. So this is what is the entire process of drug development and approval. Now, we already saw there were different, right from the target discovery, lead discovery, then optimization. You also do ADME profiling, clinical studies, and approval. So you will see that one drug, it takes up to 15 years, and at least $300 million are spent on this compound. But unfortunately, there are many reasons why the compound can fail, right? You're investing so much time and money, but there are various reasons by which the compound can fail. That can be pharmacokinetics, which you saw, right? The drug plasma concentration, very important for it to have a therapeutic effect. Animal toxicity, right? If your drug can become toxic, it has a lot of side effects. It might not have efficacy itself. That is the intention for which you want to use, that itself is not working. That is lack of efficacy. So there are various other reasons your compound can fail, and that is why it's very important that you understand the entire process. While during discovery phase itself, you need to understand and evaluate all of these reasons, so that you do not invest so much when you go forward in clinical studies, because clinical studies are even more expensive. So I also want to share with all of you, as you all know, there are small molecules like the pharmaceutical molecules you've been hearing, chemotherapy, like small molecules are used. Those are the compounds that are used. But in the late 20th to 21st century, there was a storm of biologics. So biologics, they are actually the protein-based therapeutics. So these are actually come into market from late 20th century, and you will see most of the critical diseases are treated by biologics. So the intention to share this with all of you is that biologics are becoming, the top global pharmaceuticals, and most of, at least one third of the biopharmaceutical industry in their R&D, research and development pipeline, they have biologics placed, and at least 8 of the top 10 best-selling drugs are biologics. So again, the time taken for biologics is also very long. The investment is high, but this is for your information that biologics are highly sensitive. They're very good in terms of specific treatment for diseases. So if you see in this picture, small molecule, which let's say aspirin is a small molecule, small in size. But as you move into biologics, the large molecule, which are protein based, they can be very huge. Here, it is being compared with the vehicles here so that you understand the complexity, how big these molecules are. So this is the comparison of small molecule versus large molecules, which are biologics. And if you see in the development, most of the biologics are used for cancer treatment. So all the drug development pipeline, you'll see it's the cancer related conditions. The more number of biologics are discovered, infectious diseases, and then comes autoimmune cardiovascular disease. So by disease categories, the different biologics that has been discovered. And if you go by product category, right, types of biologics or the protein therapeutics, it can be monoclonal antibodies, it can be vaccines, it can be hormones or proteins, it can be cells itself, right, cellular therapy, gene therapy. So these are different types of biologics or the proteins that are used in the treatment. So if you look into the drugs worldwide based on 2022 projections, you will see that for COVID-19 vaccine was the topmost, right? These are the leading drugs because I'm sure all of you know what was the scenario in COVID-19. So, and the second is Humira, and then you have Keytruda, and so on and so forth. So these are just to give you a snapshot of how the biologics are doing well in the market. And also very important is biosimilars, which is called. So since you see that there's so many biologics, they've been patented, right? When companies discover these protein molecules or therapeutics, they have been patented. So once the patent expires, you can actually produce any kind of molecule, a similar molecule, which is called biosimilars. So as the patents are expiring, there are a lot of different companies or the same company who are coming up with biosimilars. You will see here again in this picture that biosimilars are again in development for every original molecule, that is, it's called reference biologic or a biologic. There is a biosimilar that has been developed. So another important information which I want to share with all of you is the drug application review process, right? So I'm sure when I explained in the drug development process, once you finish phase 3 trial, you need the drug to be approved by the FDA. So there are different types of application process the FDA will review for the drug to be approved. So there are 5 different processes. So mainly for the small molecules, are the top 3, that is traditional 505(b)(1). And then if you already have a drug discovered for a particular disease but you want to use it for another disease, let's say repurposing, or you want to expand its application, then it's another application that you need to file, that is 505(b)(2), which is important for it to be considered. And NDA, abbreviated NDA, this application is mainly for bioequivalent drug. Let's not go into details. I feel that would be too much for you. But just want to tell you that bioequivalent drugs is this application. And for biologics, which I just talked about, it is the BLA application, that is section 351A, and for biosimilars, which is 351K. So these are the different review processes which the FDA would want you to apply when you discover the drug. And apart from that, there are 4 expedited approval processes, which is mentioned here. So I'll just quickly go over that. That is, if you want to fast track any drug, right? So you want to do fast track, expedite the review. because you want to treat a certain serious conditions or fulfill an unmet medical need, then you can fast track that application. So the intention is to bring the therapy as early as possible by FDA to the patient so that the patient get the maximum benefit in time. So that is why it's called fast track process. Then you have breakthrough therapy, a process which designed to expedite the development and review of drugs which may demonstrate a substantial improvement over already available therapy. Right, so there might be already another therapy, but you have discovered something that is much better than the previously available. Then you can go through the breakthrough therapy approval process. Accelerated approval process, here you can actually get approvals based on the surrogate endpoints. This is called accelerated approval. So surrogate endpoints are the ones which is much before your actual endpoint, right? The primary endpoint. Let's say you want to evaluate your drug actually prevents the death in cardiac patients. So that becomes a very later endpoint when you try to evaluate. But can there be any other endpoint that can be more upstream of that? So those are called the surrogate endpoints. Priority review here in this process, here it makes FDA to look into your application within 6 months one as you file. So that's called priority review. So these are the four different ways where you can expedite the approval processes. Apart from that, there are very many other incentive programs that FDA has provided. For example, Orphan Drug Act, GAIN, and PEPFAR. So these are different drug incentive programs. So the objective of having the expedited approval process and the intensive program is that the patient receive the drug as early as possible, especially in serious conditions and for the unmet medical need. So, what I want to, I think we are almost towards the end. What I want to share with you all is the Keytruda. So, it might be too complicated for you to understand, but I just want to help you all understand by seeing that you have a immune cell. I'm sure T cells. Everybody knows your body has immune cells. So one such type is T cells, and you have cancer cells. So you want your immune cell to kill the cancer. And how can that happen? So you will see here that when the cancer cell has this particular ligand, that is PDL1, and T cell has PD1, when these two interacts, then this cancer cells is not killed. So you want to block this interaction either with an anti-PD1 or anti-PDL1 antibody. So when you have this protein therapeutics, which is, in this case, it is a monoclonal antibody. So you block this interaction so that the cancer cells get killed. So Keytruda is one such blockbuster drug which is used for various purposes. The FDA gave so many approvals, which you will see here. It was earlier approved for advanced melanoma. Later, it was approved in 2015 for non-small cell lung carcinomas. Then came the head and neck squamous cell carcinoma, and also for Hodgkin's lymphoma. So you will see that the drug, once it was previously approved for one particular indication in cancer, it was later approved for various other indications. So this way, it's a blockbuster drug which had multiple applications. And if you see, these are the development timelines for Keytruda. I'm not going into details, but you will see that there were so many approvals for Keytruda. And another thing which I want to share with all of you is, if you go into this particular website, which is called clinicaltrials.gov, so all of you can make note, clinicaltrials.gov. Here you will see all the trials, clinical trials that is happening in the world is listed here. So if you're interested in any particular drug and any particular disease, you can go to the clinicaltrials.gov and see what kind of drugs are being tested in clinical trials, or what kind of diseases are being evaluated. So this way you will have more information on different clinical trials and the new drugs that is being discovered. Similarly, for Verquova, you will see that this is actually used for heart failure, chronic heart failure. And this drug is all similarly, you'll get more information in clinicaltrials.gov. Let's quickly understand how it works. So you will see that in cardiac patients, if there is endothelial dysfunction, the GTP gets converted into CGMP, right? If it doesn't get converted, there is a deficiency of CGMP, that is the low levels of CGMP, which leads to vascular dysfunction or myocardial dysfunction, right? The heart failure happens. So what you want to do is you want to increase the level of CGMP so that there is no myocardial or heart failure. So in that case, it is this very what is used, that is again Verquov. So what it does is it directly stimulates this guanine cyclase, right? We just saw this guanylate cyclase stimulator. So it stimulates this particular enzyme and it converts GTP into CGMP. So this level is maintained and hence you will not have any cardiac failures. So this is how the drug works. This is the mechanism of action of the drug. And if you look into again the clinical trials.gov, there are so many clinical trials that has been done. One such example is Victoria trial, that is phase 3, and it was approved in January 2021, and post-market surveillance is already ongoing. So I think, yeah, we have covered most of it today. We learned right from drug discovery, drug development, different examples of how clinical trials are done, and how the biologics have changed the field in biotechnology, how the drug development pipeline of various pharma companies are filled by biologics, and those are the information which I wanted to share with all of you. I hope this has been helpful. You will be able to use this for your studies or in your future, let's say, future steps that you're going to take for your career. Hope this concept and information will be useful. Thanks everyone for listening, and I will end my talk with this slide. Thank you so much. Sir, can you hear me?
Madam, if you want, you asked 2 questions to students, ma'am,
Okay.
on today's topic.
You're saying there are 2 questions?
So, and send through WhatsApp also.
Okay, I am not able to hear you, sir. Let me see if I can.
So after end of this session, you send through WhatsApp for me.
Madam, 2 questions.
Okay.
You type and send it in the WhatsApp, madam.
Okay.
Yeah, to Sujagowda.
The 2 questions? Sure, I can
Yeah.
send the answers.
Thank you.
Thank you so much, sir. Thanks
Yeah.
everyone for listening. I hope
Okay,
this
one
was
second.
useful. I will share my answers. Sorry for the technical glitch that I'm having, but I will share all the answers with you.
I hope there are no questions from the participants.
Is the question in the chat box mentioned? No?
No, madam. Okay, yeah. At the outstretch,
Okay, I don't see
if
questions in the
any
chat box, but if you
participants
could.
have questions, please feel free to raise your hand so that we can enable you, and you may speak to expert directly. Another 2 minutes. We'll wait for 2 minutes, madam.
Yeah, sure.
Nobody is raising, I guess. It's busy. But so if they have questions, they will contact you later, madam. to
Okay,
your
sure.
mail
No
ID.
problem.
If they send any questions to us, we will forward it to you.
Okay,
So
sure.
into the last part of the session, that is Arpana. Thanks, word of thanks. So I thank Dr. Kirthana Sindhe for producing a wonderful topic on drugs And it was a vast, it is a vast subject, to be frank. But whatever is possible, that doctor has delivered to us. Thank you very much on behalf of KSTA, our chairman S. Ayyappan, CEO A.M. Ramesh, administrative officer Madhav Gowda, scientific officers Anandar, Umesh Gadge, and Srinivasu. I thank you very much, madam, on behalf of myself too, and Sindhe Gowda too, and all the participants. I thank you for your valuable time spending with us in today's webinar. So thank you, one and all. Thank you, everyone.
Thank you so much, sir. Have a great day, everyone. Thank you.